A very good afternoon to all of you. Myself Dr. Pooja, and today we are going to discuss grand test number 182, which is actually PGI mock test. Now all of you are familiar with the pattern of the questions asked in PGI. There are multiple answers to a particular question. So always remember, if you know the subject thoroughly, then only you can attempt all the questions. So always consider this thing in mind, thorough knowledge of the subject, and you should see. This is, suppose if I talk about a particular question, if there are 4 options, there are chances that 4 options can be the answer. So you have to take very important precaution to attempt these PGI questions. Now let's start with the biochemistry question. Question number 79. Now see, which substrate is or are is used to provide energy for the body? Now the options are ketone bodies, glucose, free fatty acids, creatine phosphate and collagen. Now all of you know ketone bodies are very important, yeah, you can say they are the ones which gives energy during fasting or starvation. Or you can say brain and heart have to depend on ketone bodies for the energy. So ketone bodies are giving energy. Second option is creatine phosphate. All of you know this creatine phosphate is the energy reservoir of the muscle. Creatine in the presence of CPK enzyme forms creatine phosphate, which is giving energy for the muscles to contract as well as relax. Free fatty acids, obviously free fatty acids undergo alpha oxidation, they undergo beta oxidation, they undergo omega oxidation to form either acetyl CoA or propionyl CoA. So these are again giving energy. Options are what? Ketone bodies, yes, they give energy. Glucose, yes, they give. Glucose gives energy. One molecule of glucose undergoes glycolysis. Basically, glucose is what? It is the chief substrate for oxidation. Free fatty acids they will give. Creatine phosphate it will give. So the answer to this question is collagen won't give. So the options would be A, B, C, and D. Then question number 80. True about DNA polymerase used in PCR. Now see, PCR is what? PCR stands for polymerase chain reaction, and it is the exponential amplification of DNA. What are the prerequisites for PCR? It requires double-stranded DNA, primers which undergoes annealing, and the process takes place in the presence of heat stable DNA polymerase. And this heat stable enzyme, heat stable means because the PCR cycle runs at a very high temperature. So the enzyme which we are adding to basically polymerize DNA to amplify the amplicons, it has to be heat stable, and it is always synthesized or produced from the bacteria. Obtained from virus? No. Obtained from bacteria? Yes. Used for joining the 2 strands of DNA? It is heat stable and it adds nucleotides. So it cannot be obtained from virus. And the most important DNA polymerase which is nowadays used is, it is synthesized from Thermus aquaticus. So the answer to this question would be true is obtained from bacteria. It is heat stable and it is adding the nucleotides. Then question number 81. All are urea cycle enzymes except. Now this is again a very important question. We'll take up urea cycle. See, सबसे important thing you should know is in urea cycle it is NH2 C double bond O NH2. This comes from aspartate. This comes from carbon dioxide or bicarbonate ions, and this comes from ammonia. Right. So these are the sources of carbon and nitrogen. Urea cycle consists of 5 steps. First and second, yeah, 2 steps you can say, first and second occur in the mitochondria. Third, fourth and fifth occur in the cytosol. A rate limiting enzyme of urea cycle is CPS1, and the substrate of CPS1 are bicarbonate ions and ammonia. Now what happens is bicarbonate ions and ammonia in the presence of enzyme CPS1, Carbamoyl-phosphate synthetase 1 forms Carbamoyl-phosphate. This Carbamoyl-phosphate in the presence of, yeah, you can say with ornithine forms Citrulline. The enzyme here is ornithine transcarbamoylase. This citrulline combines with aspartic acid forming Argininosuccinate. This Argininosuccinate gives out fumaric acid. This is actually a link reaction also. Fumaric acid is an intermediate of TCA cycle in the presence of enzyme Argininosuccinase, and here the enzyme is argininosuccinate synthetase because it requires ATP. ATP is required here also. This argininosuccinate will form arginine, and this arginine will give out urea in the presence of enzyme arginase. Now see here, these first and second step occurs in the mitochondria, and third, fourth and fifth step occur in the cytosol. One important thing, CPS being a rate limiting enzyme is allosterically modulated by NAG, N-acetyl glutamate. So these are some of the very important points of urea cycle. Now let's look at the question. Question says all are urea cycle enzymes, ornithine transcarbamoylase. Just now I told you transfer of carbamoyl group from Carbamoyl-phosphate to ornithine to form citrulline. So this is correct. Carbamoyl-phosphate synthetase 1. This is a rate limiting enzyme which is combining carbon dioxide and ammonia to form Carbamoyl-phosphate. So it is an enzyme. Argininosuccinase, fourth enzyme of urea cycle, which is splitting argininosuccinate to form arginine. Citrulline synthase is not an enzyme of urea cycle because synthesis of citrulline takes place by ornithine transcarbamoylase. And argininosuccinic acid synthetase, this is the fourth enzyme which is catalyzing the synthesis of argininosuccinate in the presence of citrulline and aspartic acid. Now here one more point I would like to mention. This OTC, ornithine transcarbamoylase, is absent in the brain. Absent. And this arginase enzyme is absent in the kidney. That is why we say urea cycle is operative only and only in the liver. This is very important again. Due to the absence of ornithine transcarbamoylase enzyme in brain and due to the absence of arginase in the kidney, urea cycle is strictly operative in the liver, where 2 steps are mitochondrial and 3 steps are cytosolic, right? So the answer to this is Then 82 non-polar amino acids are. Yes, this is again a very important question which is frequently asked in the exams regarding the classification of the amino acids as per the charge on them, as per the polarity in them. Now non-polar, see, polar means which has an affinity towards water. Non-polar means which doesn't have any affinity with water. Now whether they are acidic, whether they are basic, whether they have hydroxyl group in them, whether they do not have a hydroxyl group in them, according to this, these amino acids are classified differently. Now what happens here is, if we talk about polar amino acids, polar, acidic, they are what? Aspartic acid, aspartate and glutamate. If we talk about polar basic, they are lysine and histidine. Now here the question is non-polar. Now see, let's look at the question options only. If we talk about proline, proline is a non-polar amino acid. Lysine, lysine is polar. Isoleucine is again non-polar. And arginine and asparginine will come under polar but uncharged. So you can say in this question number 82, the answer would be proline and isoleucine, which are non-polar. Other non-polar amino acids apart from lysine and isoleucine are alanine is there, valine is there, methionine is there. They will come under non-polar amino acids. Then question number 83. True statement about magnetic activated cell sorting. Now see, it is clear from the word itself. Magnetic activated cell sorting. Sorting means we are sorting out specific cells from a mixture of cells. Right? This is again a very important application which is used nowadays. Now it is based, either we can centrifugate the sample or we can put a fluorescent dye so that whatever you can say particles are there, or you can say specific particles are there, they will be fluorescent. The non-fluorescent will be what? Jinme there is no affinity between the molecules. So here it is a method to separate, yes, it is a method to separate the specific cells from a given mixture. Fluorescent dye can be used, fluorescent dye cannot be used. But overall, if we see, magnetic activated means when there is the antibody is coated with the nanoparticles and it is used to, because it is highly specific in action and they act as surface markers. So true statement about these is, yes, it is a method to separate specific cells. Antibody coated magnetic nanoparticles are used. And obviously antibodies have to be specific for certain surface markers. Otherwise there won't be any binding only. And magnetic field to some extent can be applied. But here it won't be applied. So the answer to this would be option A, option C and option D, not B and A. Reason being, I told you that magnetic activated cell sorting is further of two types, MCAS. Either it can be based on centrifugation or it can be in the presence of fluorescent molecules. But this is generally preferred because just now I told you that when fluorescence is there, we can easily, one molecule is combining say with the other molecule, there is affinity in this, and this affinity can be detected with the help of the fluorescent tag. Right? Then next question, epimer combination is or are. Now what are epimers? Now see, there are basically two things, anomers and epimers. Anomers always differ at C1 and epimers always differ at any other position except C1. Just suppose anomers, the structure of this anomer is say alpha D glucose and beta D glucose. Here you can see the difference is only at carbon 1. Difference will be only at carbon 1. So they will be linked as anomers. Epimers are what? There can be a difference in C2, there can be a difference in C3, there can be a difference in C4. So these are what? These are epimers. An example of epimers are, let's say mannose is a C2 epimer of glucose. Similarly, galactose is a C4 epimer of glucose, because C4 means they are differing in C4 position and C2 is they are differing at C2 position. Now let's look at the options. D-glucose, D-fructose. Glucose and fructose are linked as isomers. Mannose and tallose, isomers. Glucose mannose, epimer. Glucose glucose, no. Galactose and glucose, epimers. So the epimer combination is option C and option E. Next question number 85, method to determine protein structure is or are. Now protein structure, as such protein is nothing but it is a polypeptide which is formed of different amino acids. So whenever there is a structural organization of proteins, means primary, secondary, tertiary and quaternary, right? Primary depicts the linear sequence of the amino acids in a protein. Secondary is intramolecular hydrogen bonding is there, intermolecular hydrogen bonding is there. Tertiary is what? The number of bonds increases. Means tertiary is considered to be the most stable form of any protein. And quaternary is what? There are formation of different oligomers. Now see here, X-ray crystallography is a very, very important technique to determine the protein when it is in tertiary conformation. Because all of us know that enzymes are basically proteins. Whenever we have to see the active site of a protein, we have to study the structure of this by this X-ray crystallography. NMR spectroscopy, again a spectra is there, nuclear magnetic resonance, due to zwitterion structure of this. So this is again one of the very important method. Electrophoresis and ultrasonography, these two are the techniques which cannot determine protein structure. Electrophoresis can quantify it, but it cannot determine the structure of a protein. Similarly, IR spectroscopy, it is again one of the very important technique to determine the structure, whether the protein is in primary conformation, whether it is in secondary conformation, whether it is in tertiary conformation. So you can say in 85 question, A, B and E. In 85 question, X-ray crystallography, NMR and IR. These are the three very important techniques which can determine the protein structure. And another very important thing which I would mention here is protein is best viewed when it is in tertiary conformation always, due to the presence of hydrophobic bonds in this. Other bonds are ionic bonds are there, disulfide bonds are there, van der Waals bonds are there, but सबसे important is hydrophobic. That is why tertiary has a tendency to make a domain type of structure. Only the active site will be protruded out. Then question number 86. True about Gaucher's disease. Now Gaucher's disease is what? It is one of the very, very important sphingolipidosis. It is due to the deficiency of the enzyme glucocerebrosidase. And obviously if this glucocerebrosidase enzyme is absent, there will be accumulation of glucocerebrosides. So the options here is due to the deficiency. Sphingomyelinase deficiency leads to Niemann-Pick disease. Foam cell deposition is not a characteristic feature of Gaucher's disease, but pus staining is positive in this. So in Gaucher's, you can say it is due to the deficiency of enzyme glucocerebrosidase. Deposition of glucosyl cerebride will be there, and this PAS staining will be positive. So the answer of 86 question would be B, C and E. The next, after 50 grams of glucose is feed orally, there is. Now see, whenever we talk about the metabolism of glucose, we have to see there are two different cycles. One is well-fed cycle, या feed cycle of glucose, and second is the fasting cycle of glucose. Now obviously, you know, whenever we study about the metabolism, there are certain processes which lead to utilization of that glucose. There are certain processes which lead to formation of glucose. Whenever glucose is in excess, use of glucose will be there. Whenever glucose is in scarcity, definitely those processes will be accelerated which will lead to formation of glucose. Now here it is 50 gram of glucose is fed orally. Simple, there will be decreased gluconeogenesis. It's clear, gluconeogenesis cannot increase because we are already imparting glucose from outside. See, what happens is, when there are two conditions. This is feed cycle, this is fasting. Feed cycle, when the concentration of glucose is more, liver will do what? Increase in glycolysis, increase in glycogenesis, increase in lipogenesis, increase in protein synthesis, but decrease in gluconeogenesis. On the other hand, starvation, fasting mein what happens is decreased glycolysis, decreased glycogenesis, this process will decrease, this process will decrease, but lipolysis will increase because cells need energy. Energy will come from the degradation of fatty acid. Similarly, increased glycogenolysis will be there, right? And increased gluconeogenesis will be there because glucose has to be synthesized, and gluconeogenesis is accelerated during prolonged starvation. But now here if we see the question is what? When 50 grams of glucose is fed orally, then automatically there will be decreased gluconeogenesis. Because lactate production and ketone bodies play a very, very important role in starvation, right? Then question number 88, which is involved in the processing of post-transcriptional modification of messenger RNA. Now see, the RNA so produced from DNA, it is known as a hnRNA. hnRNA means heterogenetic RNA. It has two types of sequences, exons and introns. Introns are the intervening sequences and exons are the coding sequences. Now this hnRNA has to be converted into messenger RNA before it undergoes translation. And this process of conversion of hnRNA to messenger RNA is post-transcriptional modification. This is a very, very important process. Once this mRNA is produced in the nucleus, it is released in the cytoplasm so that it can undergo translation. Now what are the basic features of this post-transcriptional modification? Poly G capping at the 5' end, this is by methyl transferase enzyme. Poly A tailing at the 3' end. So basically we are marking 5', 3', GGG here and CCC here, AAA here, just to protect this hnRNA from the attack of different nucleases enzyme, and then actually it undergoes post-transcriptional modification. Now in this case ribozymes have a very important role to perform, and this ribozyme is the only enzyme which is not a protein. This is again a very important question asked in the exam. In this, transfer RNA has a catalytic activity, and this ribozyme can exhibit its function in two ways. One is simple splicing introns, or in code these are also known as self-splicing introns, and spliceosomes or spliceosomal introns. The difference between self-splicing and spliceosomal is self-splicing means introns are doing their function, and here SNRPs are used, snRNA is used here, and here introns are used. They are group 1 and group 2. As such, no classification of the groups is there. Third important point about this is, in group 2 self-splicing introns, lariat formation is there. Lariat means this type of intron come out, and here simple lariat formation is there. This is in eukaryotes also, this is in U as well as prokaryotes. Every intron, every ribozyme has 5' splice site, 3' splice site, whether it is cutting from the 5' end, whether it is cutting from the 3' end. Now let's look at the options. Lariat formation, yes, it is a feature. Splicing, it is known as splicing because spliceosome is there. Capping at 3' end, poly A formation at 5' end. These are not the options. Why? Because poly G capping is there and poly A tailing is there. So the answer to this is C and D. The next 89 question, true about IgM. Now this is a very simple question. Fixes complement, yes, it increases. This is the first antibody which increases in primary response. It has two regions, FAB and FC. FAB is antigen binding region and FC is crystallized fragment. So true about IgM are A, B and C. FC is considered to be the crystallized fragment area which is actually responsible in complement fixation. Right? So the answer to 89 question is A, B and C, not D. Question number 90, true about genes. Now all of us know genes are the smallest segment of DNA which are actually present on the chromosomes and which are transferred from one generation to another. So these are the smallest functional unit of genome. They are capable of independent expression. Promoter and enhancer genes are typical example, yes, because DNA is converted into RNA, RNA is converted into proteins. So ultimately it is what? The genes which are being transferred by transcription, by translation. So promoter are the ones which are accelerating the rate of transcription, and Cistron is the single functional unit. So the answer to this question is A, C and D. Then question number 91, true about DNA structure. Now all of us know that DNA ka structure was given by Watson and Crick. It is also known as Watson and Crick model of DNA. They were awarded Nobel Prize also for this. Watson and Crick along with Wilkins. Wilkins was the scientist who gave the X-ray crystallographic structure of DNA. Now DNA has, it is right-handed, 5 types A, B, C, D, Z. The only difference is regarding the base pairs. If we see all the 5, Z is left-handed, apart from this all are right-handed. Depending upon the base pair, just say if we talk about B DNA, it has 10 base pairs per turn, Distance between successive base pairs is 3.4 angstroms. Diameter of the pitch is 20 angstroms. Right? Now the options are what? Purines are adenine and guanine, pyrimidines are uracil and cytosine. Watson and Crick discovered structure in 1973? No. Deoxyribose-phosphate backbone with bases stacked inside? Yes. Mainly consists of left-handed, mainly it consists of right-handed helix. 5 prime, 3 prime phosphodiester? No. Phosphodiester bonding is present between 3 prime to 5 prime. Right? So, cytosine. Because you know, see, the answer to this question would be C. Students can be confused by seeing the A option. A option may you can see purines are no doubt adenine and guanine, but pyrimidines are uracil and cytosine. The major difference between DNA and RNA is RNA has uracil and DNA has thymine instead of uracil. It was discovered in 53. 3 prime to 5 prime phosphodiester bonding is present. So out of these 4 options, only the C option is correct. The backbone is, the bases are stacked inside. We have to visualize DNA as a 3-dimensional helix. Then question 92, true about hybridoma. Hybridoma technique is the most widely used technique to basically form the monoclonal antibodies. And basically what happens is, we have to make that hybridoma by taking B cells and fuse them with the antigen in the presence of polyethylene glycol, and they are multiplied in hat medium. Hat medium is hypoxanthine aminopterin thymidine medium. So some will be HGPRT positive, some will be HGPRT negative. B cells individually will die, only the hybridoma cells will survive. Why? Because otherwise what happens is, hat medium is there, aminopterin, which is basically an inhibitor of dihydrofolate reductase, will not allow the biosynthesis of purines. So here the options would be Immortalise myeloma cells, yes. Hybridoma cells produced by fusion of T cells, no. Hybridoma cells are always produced by fusion of B cells in myeloma. Cell is of human origin, no. Prior immunization is done, yes. And yes, it produces monospecific and monoclonal antibodies. That is there. That is why this hybridoma technology is carried out. So the answer to this question would be, true about hybridoma is A, D and E. Then next question, miRNA synthesis involves. Now mi is microRNA, which is basically synthesized by RNA polymerase 2 and drosha. These two are responsible for the synthesis of miRNA. Next question, restriction endonucleases cleaves. Now see, restriction endonucleases means REs. These are also known as the molecular scissors. Molecular scissors. They are highly specific in action. These are the molecular tools for recombinant DNA technology or RDT, and they are highly specific in action. If it has to cut at GATC, it is going to cut at GATC only. And when RE is cut, basically what happens is, this is the principle involved behind RDT, recombinant DNA technology. What we do is, we isolate the DNA or gene of our interest. It is not possible, the gene of interest which we are taking out, it has its origin of replication. For that we carry out the, you can say, reaction between gene of our interest and the vectors. Depending upon the number of base pairs of the gene, we select the vectors. Whether they are plasmids, whether they are cosmids, whether they are phagmids, whether they are YACs. So what we do is, this is now known as the chimeric DNA or recombinant DNA, and it is then it enters in the host cell for multiplication, and then a genomic library is there and we have to screen. Now see what happens is ki REs are always acting on double stranded DNA, always. Question is what? Yes, it is clear, double stranded DNA. Right, it cannot act on single stranded, it will act on double stranded DNA. Then 95 question, hyaluronic acid is composed of. Now see, hyaluronic acid is what? It is present in the bacterial cell wall and it is nothing but a mucopolysaccharide which has N-acetyl glucosamine and glucouronic acid. The repetitive units of this. So answer is what? It is composed of N-acetyl glucosamine and glucouronic acid. There are mucopolysaccharides have a wide classification. Some are sulfate containing, some are sulfate free. Some have glucouronic acid, some have idouronic acid. Some have only glucosamine, no uronic acid is there. For example, keratin is there. Keratin sulfate is there, chondroitin sulfate, dermatin sulfate. These are all the examples of mucopolysaccharides. But hyaluronic acid has N-acetyl glucosamine and repetitive units of glucouronic acid. So the answer to this question is A and C. Then question number 96. In Benedict's test, a red color is produced by. Now see, this Benedict's test, distinguishes between reducing and non-reducing sugars. Reducing sugars are those sugars which have a free anomeric carbon atom. And non-reducing sugars are what? Which do not have free anomeric carbon atom, or C1. Right. Now if we see glucose, fructose, galactose, mannose, lactose, starch, all are reducing sugars. Only sucrose is a non-reducing sugar. Now why sucrose is a non-reducing sugar? Because in sucrose the reaction is between the functional groups, or you can say the functional group in sucrose are hidden. Sucrose is a disaccharide of glucose and fructose. The reaction is between the functional groups on glucose and fructose. That means no carbon atom is free in that, C1 is not free. So sucrose is a non-reducing sugar. Now Benedict's test is based on, the principle behind this is enodiol formation. These enodiols are very good reducing agents which combine in the presence of this Benedict's reagent. Benedict's reagent is nothing but it is alkaline copper sulfate. Whatever enodiols are being produced at the reducing end, they are going to combine with copper sulfate, giving out a coloration. And this Benedict's test is also known as semi-quantitative test. Why semi-quantitative? Because there is a change in the coloration. Start from yellow, green to red. We say if the color is red, if it gives brick red precipitates, that means the amount of sugar is more than 2%. So it varies from 1 to 1.5, 1.5 to 2, and finally if it is more than 2%, the color would be red. Now see, the options are what? In red color is produced by sucrose, I told you it is a non-reducing sugar. Inositol. Inositol is not a sugar, it is a second messenger basically. It doesn't have a glycosidic bond in it. So there is no question of a reaction with Benedict's test. Fructose, lactose and maltose, these three are the reducing sugars because their C1 is free. So the answer to this question is कि Benedict's test will give a red color with fructose, lactose and maltose. Then we proceed to the last question, which of the following is a purine? Now see, purines are what? 7. Purines are adenine and guanine, and pyrimidines are cytosine, uracil and thymine. Thymine is present in DNA and uracil is present in RNA. Right. Now the question says, which of the following is a purine? Adenine and guanine. Because cytosine, thymine and uracil are what? They are the pyrimidines. And uracil present in RNA and thymine present in DNA. So here we finish up with the discussion of Grand Test 182 PGI. All the questions are clear to all of you? So all the best for your forthcoming exams. Bye-bye.